The overall goal of this procedure is to treat zebrafish embryos with nitrite and determine its teratogenic effect.
This method can help answer some of the key questions in the field of developmental toxicology, such as assessing the teratogenic effects of chemicals on embryonic development.
The main advantage of this procedure is that it uses zebrafish embryo, which developed externally and can be observed under a microscope.
For the experiments demonstrated here, maintain wild-type zebrafish strains, such as the Tübingen strain, at 28.5 degrees Celsius and a light-dark cycle of 14 hours and 10 hours respectively. The night before harvesting eggs, add fish water and dividers into mating tanks and place male and female fish on separate sides of the divider. To ensure that enough eggs will be produced, set up 30 pairs of fish. The next morning, after the lights turn on, remove the dividers to initiate mating. Check the mating tanks for eggs every 15 minutes. Once the fish lay eggs, use a tea strainer to harvest the embryos and combine them all into a large container with E3 buffer. Incubate the eggs at 28.5 degrees Celsius. At 1.5 hours post-fertilization, or HPF, under a dissecting microscope, use a plastic transfer pipette to discard unfertilized eggs that will appear opaque.